Hello and welcome to this ERC session. The ERC stands for the European Research Council. My colleague Constantina Topuridou and I, we work there. We have the pleasure of helping in order to choose the best scientists in Europe. The ERC is a body that is governed by scientists that are out there in the field, and the panels, they decide and to pick projects that are high risk, high gain. It's extremely competitive. It's around 10%. So the people that you're going to see here today are absolutely amazing, and we are very, very lucky to have them with us. I would like to introduce the moderator, first of all, John van der Oost, who is a leader in bacterial genetics group at Wageningen University in the Netherlands, where he's also professor. Over the last decade, he established a successful research line on prokaryotic antiviral defense systems, CRISPR-Cas and Argonaut. He's considered one of the pioneers of the CRISPR revolution for his fundamental work on unraveling the mechanism of CRISPR-based immunity in bacteria. And since 2018, he's an ERC grantee. He has an advanced grant that is called The Quest of the Argonauts, From Myth to Reality. So, John, please take over the session. Thank you very much.
Well, thank you very much. I hope you can hear me. So thank you very much, Ino. So what I will do today is I will give a brief introduction to the session. And the title of the session is CRISPR, What's Next? So what I will do is I will give a brief introduction. And by doing so, I will kind of introduce the three topics that the main speakers of this session will then share with you. So that's about CRISPR-Cas, it's about restriction modification, and it's about recombinases. Okay, so let's get started. So what I will share with you for the next 10 minutes or so is I will talk briefly about the arms race between bacteria and viruses. And then from biology to application, I will mainly talk about the biology, but as you will see that the topics that I will introduce that I already mentioned, the speakers will elaborate on that, and then they will also talk about applications. Okay, so every organism on this planet is facing viruses. So we all know that we ourselves suffer from viruses, but also plants. And in fact, it was in plants that viruses were defected, or they were first found in plants, and it was by a Dutch guy called Martinus Beijerinck, and it was more than 100 years ago. And then, of course, also bacteria, they suffer from viruses. I must say that each organism has rather specific viruses. So, like the coronavirus that now is attacking mankind, bacteria do not suffer from that. Okay, let's now focus on the bacteria. So here you see basically in green the virus attacking a bacterium. So the bacterium is in blue, and you see also that, and that's with this symbol, I want to indicate that also these bacteria can defend themselves against the attack. So just like in many cases, also the more complicated organisms, they develop defense systems to fight the viruses or to protect themselves. Also bacteria can do so. So the CRE system, I will elaborate on that in a minute. That's a system that viruses use to target the bacteria or to integrate in the DNA of bacteria, as I will show you. But there are also defense systems, and the restriction modification, or RM system, that has been discovered many years ago in the late 1960s. They were discovered for the first time. And the CRISPR system has only been discovered something like 15 years ago. Okay, so how does that look like? So when a virus infects a bacterial cell, so first there is this recognition, because also the bacterial virus, the bacterial viruses, they only recognize specific hosts, and that's dedicated or that's determined by the cell wall of the molecules that are present on the cell wall of these bacteria. And then when recognition appears, then the genetic material, in this case DNA, is injected into the bacterial host cell. And then two things can happen. So either the DNA, in this case, integrates into the DNA of the host, so into the bacterial chromosome in this case, or the DNA starts to replicate, and that allows then for direct proliferation of the viruses, as I will show you in a minute, and that often leads to death of the infected host cell. But let's first now focus on the integration. So several systems that are encoded by the DNA of the viruses. In this case, these viruses are called bacteriophages. On that DNA, the genes are encoded that allow for the integration in the host chromosome. And these systems, there are several systems that have been well described, like the system from the bacteriophage lambda, with the integrase and the excision enzyme that recognize the so-called attachment sites, or the ATT sites. And then there is the phage P1, has similar systems, so it's variations on a theme, but it's a single recombinase called CRI that recognizes the LOX sites. And then there are many more. But or later on in this session, we will hear a lot about the CRI-like recombinases. And then these recombinases, as I already indicated, it's not a one-way integration, but they also can leave the chromosome again, and then they start to replicate. So often when the host organism is exposed to stress, then the virus gets a signal, well, it's time to go and to leave the system before the whole cell collapses or dies. And that is what then happens. So then the replication proliferation cycle starts. And how does that look like? Well, it looks like this. So the DNA is first transcribed to RNA, translated to protein, and in addition, it's also the DNA is also replicated, and the replicated DNA is then packaged into the protein capsids, and that gives rise to intact viral particles. So typically, such a viral infection, so a single infection by a single virus gives rise to the generation of between 20 and 50 and even more new phage particles. And then the cycle can start over again until the viruses encounter host cells that developed efficient defense systems. And many of these defense systems have been recognized, and basically they operate at all levels of the proliferation cycle of the virus. So starting with the attachment, then with the injection of the DNA. Sometimes the infection leads to triggering of a dormant state or even suicide of the cell, just to slow down the proliferation process. And then the two systems that I already mentioned, so the restriction-modification system, and we will hear much more about the details of that during the third talk today. So the restriction-modification system can be considered as an innate immune system. And then, as said, the CRISPR-Cas system, that is considered to be an adaptive immune system. And again, we will hear much more about that in the second talk of today. And also, and that fits with the title of this session, What's next? There will be, or recently it became clear that there will be discoveries in the near future of many, many more defense systems. I don't know if they will be as exciting as CRISPR-Cas and as exciting as the restriction modification system, but they are definitely out there, and we will hear much more about that in the coming years. So these are the two systems that we will hear much more about today. And this is basically the summary of this presentation. So we will start today with a talk by Martin Jinek on the CRISPR-Cas system. So from adaptive immunity basically to genome editing, because we're in the middle of a genome editing revolution. Then the restriction modification system. I already indicated that should be considered as an innate immune system that has been instrumental for allowing cloning. So the cut and paste of DNA fragments, that was the basis for the genetics revolution that started in the 1980s. But also we will hear today that also these tools can be very useful for analysis of epigenetics. And then last but not least, the Cre recombinase or homologs of that recombinase. So from the phage integration that I described to you, now we will hear that also these enzymes can be repurposed for accurate genome surgery, as it is said. So with that, I will stop here. So here again, you see the three speakers that are coming. And I will start by introduce Martin Jinek. So here I stopped with my presentation and the people behind the scenes can upload the presentation of Martin. Martin Jinek. Okay, so the next speaker of this session will be Martin Jinek. And Martin Jinek was raised in the Czech Republic, and then he moved to the UK to study natural sciences in Cambridge. And after that, he moved to Germany, to Heidelberg, where he did his PhD research at the EMBL in the group of Professor Elena Conti. After completing that successfully, he moved to Berkeley, to the lab of Jennifer Doudna, and it was in 2012 where he published a milestone paper on CRISPR-Cas. And in that paper, he described the relevant details of the CRISPR-Cas9 mechanism. And that really triggered the CRISPR-based genome editing revolution. A year later, so in 2013, Martin moved to Switzerland, where he became associate professor at the University of Zurich, where his group currently studies structure-function relations of different CRISPR-Cas nucleases. And the aim is to reveal novel insights in their molecular mechanism and also look for possibilities to apply these systems as genome editing tools mainly. Martin has received several prestigious awards including ERC starting and consolidator grants in 2013 and 2018. And today, Martin will talk about CRISPR-Cas genome editors from bacteria. Please, Martin, the floor is yours.
Okay, so good afternoon, everyone. Hello, it's a real pleasure to take part in this Science Open Forum, and I would like to thank John and Ino for the introduction to this session. What I would like to do in my presentation is to give you an overview of CRISPR-Cas, tell you a little bit about where this technology actually comes from and the kind of capabilities that it has given us in being able to modify the genetic information of many other organisms and cell types. I should say that I basically come to this from the perspective of somebody who's been interested in RNA biology for a long time. As you will see, RNA plays a very important role at the core of these CRISPR-Cas systems. And basically, through the understanding of the protein RNA complexes and enzymes that are involved in these pathways, we basically hope to reveal the mechanisms and their functional implications, not just for the biology of these systems, but also for their applications in biotechnology. And so there's a lot of excitement about CRISPR-Cas technologies as something that can be deployed effectively as a programmable surgery for the genome. It's something that allows us now to modify fairly simply, fast, and with a great deal of precision the genetic information of organisms, of cells, and also potentially of human patients. And so this is turning into really a powerful method for basic research, biotechnology, and also molecular medicine. So thanks to this, we will have in the not so distant future, hopefully genetic therapies based on this technology. And it's something that I think for the first time in our own history as humanity gives us the power to really modify our own DNA and shape our own evolution, if you will. And with that power, of course, comes a great deal of responsibility to make sure that this technology is used in a responsible manner. And so the question that often comes up is where does this technology come from? How did this actually came about? And as John already mentioned in his introduction, this is something that's based on a naturally occurring system that provides adaptive immunity against viruses in bacterial cells. So it's something that bacteria have evolved in the course of evolution to basically get an upper hand in this ongoing arms race between bacteria and the viruses that infect them. And so in this way, the CRISPR-Cas system actually joins a long list of molecular activities and tools that have been basically exploited and developed based on this ongoing arms race between bacteria and viruses. And as John already mentioned, this is something that was started in the late 1960s with the discovery of the restriction modification systems and the restriction endonuclease enzymes. And so very briefly, how does the CRISPR-Cas system actually work? Well, as an adaptive system, it basically has to have a kind of a memory component. And so what bacteria actually do when they encounter these viruses, they basically steal fragments of the viral DNA and incorporate them into their own genome in a specific kind of genetic structure in which this information is recorded and can be basically passed on. And meaning that if the bacterium basically incorporates a fragment of the DNA of the virus into this so-called CRISPR locus, it will become immune to subsequent infections by the virus whose DNA basically matches the sequence of that fragment incorporated in its own genome. Now, these DNA fragments are then basically transcribed and used to generate guide RNAs, which associate with additional components of the CRISPR system called Cas proteins, some of which function as nucleases. So basically enzymes that target other nucleic acids. And in this case, this association of the Cas nuclease with the guide RNA basically provides a targeting mechanism with which the bacterium can recognize whenever a virus comes in and tries to inject its own DNA into the bacteria. And basically using this guide RNA molecule will form a complex with the injected nucleic acid and will target that nucleic acid for destruction, typically by the nuclease-based degradation mechanism. Okay, so we have basically these three steps. The viral DNA is first inserted into the bacterial genome. It's basically then used to make these RNA-guided targeting complexes, which then recognize the sort of second round of infection by base-pairing interactions between the guide and the target DNA. And then finally, the DNA is basically destroyed by one of these Cas proteins acting as a nuclease enzyme. Okay. And so basically, this is then where the translation of the system from the naturally occurring scenario where we have bacteria targeting viruses is used to basically do genome editing, because the main idea behind this is that we have RNA-guided DNA cutting enzymes that can be programmed using artificial guide RNA molecules to then target DNA in the genome of other types of cells. And in this way, by basically combining the site-specific DNA cutting and the naturally occurring DNA repair mechanisms of these cells, we can basically achieve site-specific modification of the DNA once the system has been deployed in these cells. And so we basically go from a double-strand break in the DNA generated by these CRISPR enzymes to DNA repair. And because we can, to some extent, steer the outcome of the DNA repair mechanism, we can basically then achieve modifications of the DNA. And then depending on which DNA repair pathway can be used for this, we can either generate simple types of modifications like mutations, and by introducing mutations we can basically knock out genes, or we can do more complicated types of genetic manipulations where we can insert sequences and we can even correct mutations or overwrite parts of the genetic code in the cells. And so put very simply, the CRISPR-Cas as a genome editing technology is really very similar to essentially the find and replace function in a text editor, where basically we have an algorithm that's based on a 20-letter search string that allows us to find parts of the text in the genome and then insert, delete, or overwrite parts of that genetic sequence. Now, in my own laboratory, my research is really focused on understanding how these CRISPR-Cas nucleases that have been now repurposed as genome editors actually work. And this, as John already mentioned, is in many ways based on some of the early work that we did together with Jennifer Doudna in the United States, where we discovered that basically there are these RNA guided DNA cutting enzymes, and that they can be engineered in a way that we have basically a programmable one protein, one RNA system, where all we need to do to target the nuclease to cut the DNA in a specific place is to provide a single RNA molecule that will be targeting a specific DNA sequence, and whose sequence basically matches the sequence of the guide RNA. Now, in my own lab, since those days, we've been trying to understand how this system works in three-dimensional space by studying the structures of these molecules. And this is just an image of a 3D printed model that's based on one of the molecular structures that we determined using X-ray crystallography, where we basically see in white, this is the Cas9 nuclease. In red, this is then the guide RNA, which, as you see here, is making an RNA-DNA duplex with one of the strands in the target DNA. And so this is basically how these enzymes then attach to the correct site in the DNA to then catalyze the cutting of the DNA. So in my lab, we've been basically trying to understand the structural and mechanistic aspects of these enzymes. We started with Cas9 already quite some time ago. More recently, we have focused also on one of the other CRISPR-associated enzymes called Cas12a, which actually started as a collaboration with John van der Oost's group in the Netherlands. And so we have now basically these enzymes building up the molecular toolkit for genome editing. And so how can we now use these tools for human therapies? Well, the idea is that by basically programming these tools to recognize specific sites in the human genome, we can deploy these to, for example, correct genetic mutations that underlie some of the well-known genetic diseases in humans. An example of this would be sickle cell disease, where a single nucleotide, a single letter mutation in the DNA that corresponds to the gene that encodes one of the components of hemoglobin, basically results in the production of a misfolded or misfunctioning protein. And so using CRISPR-Cas, we could potentially one day use this in a therapeutic strategy that aims to correct this mutation and revert it back to its healthy version. Okay, so the idea here would be to use these tools for gene correction, and ultimately one could consider CRISPR-Cas as a tool for personalized genetic medicine because of its programmability. And to deploy CRISPR-Cas in human patients, there are several different scenarios what could consider. One would be, of course, to do the genome editing in the patient itself by targeting a specific tissue or a specific organ using various delivery methods to deliver the Cas9 protein and the guide RNA. Something that's more feasible, at least in the short run, will be genetic or genome editing based on ex vivo strategies, where cells will be taken out of human patients, modified in the laboratory, and then reintroduced into the patient to cure a specific disease condition. However, there are a lot of challenges in this that remain. One big question is a question of specificity and safety of these systems. It's known that they can have off-target effects, and so there's a lot of effort in the field aimed at reducing the off-target activities of these nucleases. Delivery of these systems into cells for therapeutic purposes, is of course another big challenge. And again, a number of methods have been developed recently. And finally, I guess the third major challenge is basically the idea of being able to control the DNA repair outcomes of the double-strand DNA breaks that are generated by these systems. So somehow controlling the interface between the genome editor nucleases and the downstream DNA repair processes is also something that's very important. Now, in my lab, what we're hoping is that by continuing the structural and mechanistic studies of these enzymes, we can contribute basically to addressing these issues and basically improving the technology over time to make it as safe and as specific and as efficient as possible for applications. Now, finally, what I would like to mention is our ongoing work in the context of the ERC Consolidator grant that is currently funding part of my research in the laboratory, and this is where we're basically trying to take CRISPR to the next level. As John mentioned, there's a lot of genetic diversity in the prokaryotic kingdom, in bacteria and archaea, and we're hoping to exploit some of this diversity to find new molecular tools. And so we're looking at some of the aspects of CRISPR-Cas pathways, as well as at some of the new genome defense systems that have been identified in recent years, to characterize their molecular activities and basically hoping to find new molecular tools that could be repurposed for genome editing. And so with this, what I would like to conclude with is that there's a lot of, we're only really seeing the tip of the iceberg, I think. There's a lot of unknown, uncharacterized biology out there. And if you are a bioengineer that wants to look for or wants to develop new molecular tools, and you think that you have a great idea, then it's quite likely that nature has probably been there before you. And so looking and trying to discover new molecular tools in the biosphere is something that still has a tremendous potential for the future. So with that, I would like to thank you very much for your attention. Thank you very much for listening. I would just like to briefly acknowledge the people working in my laboratory who do fantastic work, and of course the agencies that provide funding for our research. So thank you very much.
Mark studied biochemistry at the University of Liverpool, and then he moved on to study enzymology at the universities of Southampton and Bristol, also in the UK. And during that time, he worked on the elucidation of the roles of short and long-range DNA interactions by different types of restriction enzymes. And at present, Mark is professor at biochemistry at the University of Bristol, where he uses biophysical methods to study microbial defense systems. And these defense systems are both restriction enzymes, as I already mentioned, but also CRISPR-Cas. And Mark established his research group in Bristol in 1989, and it's supported by several awards and fellowships from the Wellcome Trust. And on top of that, he received an ERC advance grant in 2018. And today, Mark will talk about bacterial immune systems that target epigenetic modifications of DNA. Mark, the floor is yours.
Thank you, John, for the introduction, and thank you to the organisers for inviting me. Greetings from Bristol to everybody. So I'm going to talk about different type of system, restriction modification systems that recognize epigenetic modifications of DNA. I thought I'd just start by briefly mentioning what this means. So Martin will have touched on this idea that information in DNA is stored as a series of letters, A, C, G, and T, and these encode amino acids that then lead to proteins. But in addition to those letters, there are also these epigenetic changes, which are heritable changes in the phenotype of a cell or an organism that are not encoded in the DNA, so not encoded in that series of letters. And one example of epigenetics is modifications of DNA, and I've shown examples here. This is a cytosine base in DNA that's had this additional methyl group added to it. So it's had this DNA modification or methylation added. And this is another form that's found in human cells, 5-hydroxymethylcytosine, this additional hydroxymethyl group onto a cytosine. And so this adds an extra level of information onto the DNA. So in addition to the Cs, you also have these 5-methyl Cs. And in high eukaryotes, this can have a lot of different effects. It can change the regulation of genes. Genes can get turned on or off. It can change the structure of the genetic information in the form of chromatin. It can change how cells differentiate, how a stem cell becomes different types of cell in the body. That can be programmed by methylation changes. And you can have different methylation depending on whether the DNA came from your maternal or your paternal line. So certain alleles can get turned on or off from different lines in your genetic background based on methylation. And it can also lead to diseases as well, including cancers, and may also have a role in aging. So you have these very kind of pleiotropic effects of methylation, of very complex levels of changes. So it'd be very interesting to understand this in more detail, to have better tools to understand DNA methylation. And as well as in eukaryotes, methylation of DNA is also found in prokaryotes, like bacteria and archaea. And that's what I'll talk about in terms of restriction modification. So John already mentioned this idea that these rather beautiful looking viruses are not particularly nice for these bacteria, because the poor bacteria has the DNA of the virus injected into it. This replicates, and depending on the conditions, this can then produce multiple new viruses when the cell lyses, and you release the viruses into the environment. And so this has been happening, this interaction between viruses and bacteria, for billions of years on the Earth. And bacteria have therefore evolved various ways to evade infection by the bacteriophage, or phage as I'm calling them here. And one of these methods is restriction modification. So Martin introduced CRISPR as one kind of weapon in the armory that bacteria use, and this is another method. Relies on two different enzyme activities, an endonuclease, which I've represented by this little Pac-Man figure, and a methyltransferase. And so what happens is that the endonuclease acts as a molecular scissors. So in the same way that Cas9 can be targeted to a site on DNA, restriction endonucleases also target themselves to specific sequences, somewhat shorter sequences than the ones that Cas9 can use. So for example, AGGCCT. If that sequence appears in DNA, this endonuclease will bind to that sequence, recognize it, and it'll cut both strands of the DNA to make a double-strand break. So the consequence of that is that when this phage injects its DNA into the cell, there are multiple copies of this AGGCCT sequence. The endonuclease binds to those, and it chews up the DNA into multiple fragments, and that DNA is no longer infective. But what's going to stop this Pac-Man from attacking the host DNA? And the reason why all those AGGCCT sequences within the host DNA are not cut is because this partner enzyme, the methyltransferase, produces methylation of the DNA. So it produces epigenetic changes in the host DNA. You get these methyl groups added to these cytosine bases, which appear as these bulky methyl groups within the major groove of the DNA. And these block the binding of the endonuclease to these sites. So by virtue of being methylated, the endonuclease will no longer cut the host DNA, and therefore this system can distinguish between self DNA due to this epigenetic pattern from foreign DNA, because this phage DNA is not modified. And as I mentioned, because it's been going on for billions of years as interactions, the phage have then come up with alternative ways to bypass these systems. And one of the ways they do that is by modifying their genomes. So it might be that the phage picks up methylation of its DNA because it evaded the restriction enzyme, it became methylated, and then infected the cell, released methylated viruses into the environment. But there are also examples of phage where they've evolved metabolic pathways to actually modify the bases within their genome. So for example, some of the T4 phages have every single cytosine glucosylated, so the glucose group on the cytosines. And the effect of this is that when this phage with modified DNA infects a cell which has a restriction modification system, and there are different types, such as I didn't mention, different types of restriction modification systems, type 1, 2, and 3, and these will just have different ways of interacting and cutting the DNA. They are blocked because the sites they want to bind to, these restriction sites, are methylated. And so this modified phage can then infect this bacteria, and this obviously is going to cause problems. And so as part of this arms race, which John mentioned, between the phage and the bacteria, each trying to evolve ways to evade one another, the bacteria have evolved ways to evade these phage with modified DNA, and that's the type 4 restriction modification systems. So these little Pac-Man endonucleases also recognize DNA, but they recognize DNA that has been modified. So they don't recognize unmodified DNA, they recognize DNA which has modification. So for example, a methylated site or even a glucosylated site can be recognized, and different type 4 enzymes recognize different modifications. So this phage thinks it's going to do really well, it's going to inject its modified DNA, but unfortunately, because this bacteria has a type 4 endonuclease, it recognizes that DNA as being modified, and it cuts up the DNA. There isn't a partner methyltransferase in this system, it's simply the endonucleases, and the host DNA is not modified, and therefore it is not cut. It's not recognized as being foreign because it doesn't carry the methylation. What it's recognizing as being foreign is this methylation. So these type 4 systems were actually the very first restriction modification system to be discovered in a very early microbial work. They were subsequently purified and examined in the 1980s. We still don't know very much about their activity, both as enzymes and their activity within cells. So as part of my ERC funding, my group is trying to look at these type 4 enzymes at various different levels. We want to understand, in my group in Bristol, the enzymology and protein dynamics of these systems. How do they actually recognize these modified bases within DNA, and how does that lead to the cleavage of the DNA? And you can already go to one of the biotechnology companies like New England Biolabs, open their catalog or look at their catalog online these days, and you'll find type 4 restriction enzymes available for people who want to study epigenetics. But those enzymes aren't perfect, and we'd like to try and re-engineer them to make them slightly better and more applicable to some of the things that people want to look at. And then in addition, we have a collaboration as part of this grant with Ed Silvestra's group in Exeter. And Edzer is interested, in fact, he did a lot of his early work on CRISPR, along with John actually, and he's now moved from biochemistry into more evolutionary ecology. So Edzer is interested in understanding how CRISPR systems within actual E. coli and other bacterial cells, what advantage they give to those bacteria under different conditions, under different numbers of phage infections. And he's now moving into this field with restriction modification systems and the type 4 enzymes. So I'll briefly mention what he's doing as well. So one of the kind of interesting things about these molecular scissors, these restriction modification systems, is that if you have a modification on your DNA, a methylation site, you might expect that that would get recognized by the endonuclease, and you get a double-strand break introduced into the DNA at that site. But it turns out that many of the type 4 enzymes, and in fact, many restriction enzymes altogether, need to recognize multiple sites before they trigger cleavage. So recognition of one site on DNA is insufficient. You have to recognize 2 sites. And only when you have 2 sites, you then trigger cleavage of the DNA. But exactly how does that happen? How does the enzyme know there's a site over here and a site over here, when those two sites can be separated by tens, hundreds, or even thousands of base pairs of DNA. There has to be some kind of communication mechanism. So I'll give you one example of a type 4 enzyme to show you that these systems are quite complex. So this is a protein called MCR-BC. It comprises two proteins, MCR-B and MCR-C. And MCR-C is the nuclease. And this protein comes together, two copies of the protein to form a dimer. And you might expect that's all you need for your molecular scissors, but in fact you don't. You need additional molecular motors, and these come from MCR-B. And MCR-B has a domain called the modified DNA target recognition domain, shown in this crystal structure in blue. That recognizes the modified DNA by flipping the base out of the DNA. Here's the DNA in gray, and the modified base has actually been removed from the double-stranded DNA structure into a binding pocket within the target recognition domain to be recognized. And that domain is attached to this, what's called a AAA plus GTPase. So GTP is a chemical energy within the cell, and this protein can bind and break down this GTP and convert chemical energy into some kind of mechanical motion. And what's particularly unusual here is you've got this 6 of these MCR-B proteins come together to form a ring, and the nuclease actually inserts itself within that ring, which is kind of unusual. Often when there's holes in proteins, DNA or RNA goes into the hole, but in this case, it's another protein inserting itself inside. So you have the nuclease inserted into one ring here and a second ring here. So you can see there's actually rather large number of recognition modules on both sides of this protein. There's GTP hydrolysis going on in here, and there's GTP hydrolysis going on here, all in order to allow this nuclease to cut the DNA. So how does it actually carry out this long-range communication? So one of the things we do in my lab is we do single molecule biochemistry to try and understand how proteins work. And the principle of single molecule biochemistry is, rather than taking a test tube full of enzymes and trying to work out what that ensemble is doing, you actually look at individual enzymes working on DNA one at a time. And the way we can do this, one way we can do this, is that we attach a piece of DNA in red here to a glass cover slip. The thing in gray at the bottom is a glass cover slip. We attach one end of a linear piece of DNA to the glass cover slip and the other end to a small magnetic particle, and we can stretch that DNA using magnets above the microscope. So you can basically stretch this DNA out. We can then observe the length of the DNA, and these little blue circles represent enzymes, and we can ask the question, well, if this enzyme has to recognize multiple methylated sites on the DNA, how does it communicate between them? And one way could be that the enzyme wanders down the DNA using this GTP hydrolysis like a molecular motor. It moves down the DNA until it meets a partner enzyme and then triggers cleavage. If that was the case, the DNA length doesn't have to change because the enzymes simply move along the DNA as a track. However, another way of interacting between these two enzymes might be to form a loop in the DNA. So these two proteins come together, they loop out the intervening DNA, and then they trigger cleavage of the DNA. And in this case, the bead attached to the end of the DNA has to move down in order to form this loop. So in this graph here, there's time along the x-axis and DNA extension, DNA length along the y-axis. And we add our enzyme and GTP, and then we watch, there's two DNAs we're watching here, and you'll see it looks quite noisy, and that's because this is observing the length of the DNA, and there's Brownian motion. The bead is actually being moved around by water molecules. And these little spikes here is when cleavage occurred, and the DNA wasn't shortened. So it looks like this MCR-BC enzyme is taking this pathway, it's moving down the DNA, it's not forming these loops in DNA. However, we've been unable to actually observe this motion using any of our standard tests for molecular motors. So we're going to use a different single molecule method, which we used previously with our type 3 restriction modification enzymes, the work we did, where we can directly observe the motion of the protein on DNA by attaching a fluorescent molecule, for example, quantum dot, which is a type of fluorescent nanocrystal. And in this movie, what you're going to see is a piece of DNA which is stretched out in this direction here. And here you'll see an enzyme bound to a recognition site on the DNA. And This enzyme also requires chemical energy in the form of ATP this time. And you'll see it starts to move, it rebinds to the same recognition site it bound to, and it moves again. And what you'll notice this movement is that it's not in just one direction, it moves over here and it moves back again, it moves backwards and forwards. So what this enzyme is doing is it's actually searching the DNA to find its second site by a method called DNA sliding. It's not actually acting as a molecular motor, it's diffusing on DNA, and we think this is what MCR-BC is doing. So this very, very complicated molecular machine comprising these two double rings hydrolyzing GTP is actually most likely just randomly moving up and down the DNA trying to find two sites that are methylated. So these are clearly very, very complicated systems. But because they're modular, and I mentioned this target recognition domain, which flips out the base and recognizes it, that these are relatively simple, and we can take these domains and we can try and mutate them to change them to recognize different sequences by, for example, methods of in vitro directed evolution. Now, the type 4 enzymes tend to recognize sites which are fairly degenerate. So compared to that sequence I showed you earlier for a type 2 restriction enzyme, the type 4s recognize, for example, an A followed by a modified base or a G followed by a modified base. So rather simple sequences. So these are going to be more simple maybe to change their recognition. And one thing we'd like to try and do is make these tools better. So for example, in vertebrates, these CPG sequences are often methylated, but there's not a good restriction modification system that can actually recognize these CPGs. So we'd like to try and make a system which can recognize these, or perhaps distinguish between a methyl versus a hydroxymethyl modification of a C, or to recognize different sequences. So if you might be interested in working on vertebrates, or you might be interested in working on plants, you want to look at different methylations in different contexts. So that's one aspect of our project, is trying to evolve these. And I think Frank will talk about using kind of modular domains to build systems which can do things. And then finally, the work that EDS is doing is to ask the question, well, what is the benefit of having a type 4 restriction modification system? So for the classical type 1, 2, and 3 systems, which have this methylation, you might think, well, the benefit is that, of course, you stop phage infection, but the cost is that occasionally the endonuclease will cut the host DNA, forming a toxic break in the DNA. And the reason why this happens is that you get occasionally unmodified sites on the host DNA. They are then targeted as being foreign DNA, although they're not, and you get a toxic break in the DNA. So there's a cost involved in having a restriction modification system. For the type 4 system, which target modified phage, you might think, well, actually, it's never going to target the host DNA because this isn't modified. So therefore, there's only benefits, there's no costs. However, there's probably a cost involved in what's called horizontal gene transfer. So within populations of bacteria, genetic information is being transferred all the time between bacteria. And this is why you get problems like antimicrobial resistance, because you get genes or virulence changes in bacteria, because you get genes transferred between different bacteria. And in this case, between these two different bacteria, you can see a difference is that if the DNA comes from this bacteria to this one, it's not methylated and therefore it's going to get cut. If DNA transfers from this bacteria to this one, it's methylated, so it's going to get cut. So these bacteria can't transfer DNA very readily. Well, actually, no one's actually tested this properly. And so this is one of the things that EDSA wants to do, is try and test this idea of, given horizontal gene transfer between bacteria, what are the particular barriers that these different restriction modification systems present? And is there a particular difference in the barrier when you have a type 4 system compared to a type 1, 2, or 3? So this is sort of taking our understanding of these enzymes from the kind of mechanistic level right the way through to what they're doing in the cell, also to try and make them into better tools. So thank you very much for listening. I thank my group in Bristol, EDSA's group in Exeter, and our long-term collaborator, Ralph Seidel, who's in Leipzig, who does a lot of our single molecule collaborations with us. Thank you.
Okay, so we already reached the final presentation, and it will be by Frank Buchholz from Germany. And Frank did his PhD at EMBL Heidelberg, so also in Germany, in 1998. Sorry. After that, he moved to the United States to work for a 4-year period as a postdoc in the group of Michael Bishop, and it was at the University of California in San Francisco. After that, he moved back to Germany, where he became group leader at the Max Planck Institute for Molecular Cell Biology and Genetics in Dresden. And that was between 2002 and 2010. And after that, he stayed in Dresden, but he moved to the medical faculty of the Technical University over there. And that is where he is still professor for medical systems biology. And during his career, especially in the beginning, Frank worked on applications of RNA interference, and later on, more and more, he started to work on different aspects of recombination. And that is what he will also talk about today. And the title of his talk will be Designer Recombinases for Efficient and Safe Genome Surgery. Frank, the floor is yours.
Thanks very much, John, and thanks everyone for listening, and thanks for the organizers for the invitation to let me present our ERC project here and additional things that we are doing in the lab. Now I start my lab. We're trying to apply these things to human health, and I always like to compare the situation that we are in right now with maybe the time a couple of not so long ago actually, when surgery was entering the medical field. You have to know, or you might remember, that surgery was not a medical discipline for a long time, and in fact, when you had to go you had to go to the barber if you needed to get surgery. But a lot of things had to happen, of course, during this time to make surgery a medical discipline. And nowadays, of course, it is one of the most important medical disciplines, probably. And the transition to this was that the anatomy of the human body needed to be understood much better, so that basically the surgeon knows what he's doing, where to cut, where can he cut into the body without damaging too much. Other things, of course, also had to happen, but that was certainly one of the very important things. And that may be an analogy. We're kind of that spot right now where surgery moved into a medical discipline with genome surgery. So, and for genome surgery, genomics is the basis. So we need to know the sequence, of course, of diseases. So which diseases are there and which mutations are causing diseases, and only then can we start to think about how can we repair them. And we're basically at that situation at the moment. And to do this, just like a surgeon, of course, we need the right tools. And we have heard about some of the tools already here, some listed. There are mainly nucleases like homing endonucleases, zinc finger nucleases, TALENs, and most recently the CRISPR-Cas system, of course. And what I would like to do today is to introduce to you another very powerful tool, which are site-specific recombinases, which can also be used. And maybe to compare the two systems, one of the important things is that nucleases, as we have heard, basically cut, and they introduce a double-strand break in the DNA. And this has to be repaired. And we've heard already about cellular DNA repair pathways. So there are different ones in the cell. So the cell has to decide how to repair this. This is not so easy to steer. And very frequently, this leads to DNA modifications, so-called indels. So this process is error-prone very typically. And if you think about therapeutic applications, maybe this is not the best way to go into therapy with this. So ideally, you would want to know exactly what the DNA looks like after you have introduced a change. And here maybe DNA, the recombinases come into play. But you can think of them as scissors which bring their own glue with them. So they actually very precisely glue the ends of where they have cut back together in a form of conservative recombination, and therefore you can view them as error-free. You know exactly afterwards how your DNA is going to look like, and we think that that's an advantage of the recombinases, in particular when it comes to therapeutic applications. But of course, you've heard how easy it is to program CRISPR. You just need to basically order a piece of RNA, and because of the sequence, you know exactly where these things are cleaving. So the enzymes that actually appear in nature, they have their recognition target sequence, but this sequence is of course not found in the human genome. So one of the problems that we were facing is how do we make these recombinases to cut somewhere where we want them to cut. And what we then basically used is that we decided to use what humans have been using for a long, long time, basically breeding. So if you think of dogs, for instance, humans have, of course, done this for a long, long time to breed small dogs into big dogs. And basically, we apply the same technologies by crossing different recombinases and thereby generating new varieties to make enzymes which have a new specificity. So we use the power of molecular-directed evolution to make new recombinases. Just as a side notice, this process, of course, just 2 years back has been acknowledged with a Nobel Prize to be able to basically use directed evolution to make new enzymes. And the first application that we used this for, it was HIV. This is a couple of years back. So if you remember your HIV biology, it's a virus again that this time infects humans, of course. It's a retrovirus, and thereby it integrates its own genome into the host genome, into the human genome. There are very good therapies today. So by basically inhibiting different pathways of the virus, here indicated with red arrows, for instance, the reverse transcription or the budding or the translation, by combining different drugs, we have been able to really live with this disease quite well. The problem, though, is that as it is a retrovirus, it integrates its own genome into the genome of the whole cell. And therefore, we have effective suppression of the virus, but no cure. This is why patients that have been infected with HIV today have to take medication for the rest of their life. So every day they have to take pills and to suppress that the virus doesn't take over and kill the cell. So we came up with a different idea in that potentially is really a curative approach by attacking the integrated provirus. And the way that we envision that is basically shown in the movie here. I don't know, it should be playing automatically. It's not. So probably, I don't know if we can go back.
Should I try?
I can make it go back. You can, yeah. Well, otherwise, I just explain what would happen. So what you see here is the viral DNA that has integrated itself into the genome, and it's flanked by these red sequences. This is how the virus integrates. They're called long terminal repeats. And what we managed to do is we managed to make a recombinase which can recognize these sequences in this red part of the viral genome and excise the virus from the genome, and thereby really gets rid of this virus and thereby offers a totally new way of combating this disease. And we've actually moved this further and have developed a potential therapy that is going to be tested in humans. Actually, we've moved this all the way towards clinical testing. We have obtained funding to do a first phase 1b/2a clinical trial, where the plan is to take infected patients, bring the recombinase viral vector into the hematopoietic stem cells of these patients, give these cells back to the patient, which is now equipped with a recombinase, and when their cells get infected, is able to basically heal themselves, the cells can cure themselves by cutting out the integrated provirus from the cells. So that's a therapy that we will test hopefully soon. We, of course, it didn't stop there, but we wanted to know, can we use these site-specific recombinases for other potential applications? And actually, recombinases are pretty versatile. I've shown you an example where they can cut something out, but they're also actually able to invert a sequence. So if the target sites are oriented with these little triangles here in an inverted orientation, they can basically turn a piece of DNA around on the chromosome. And now the question, of course, is, are there any genetic diseases that are caused by inversions? And I wouldn't tell you this if it wouldn't be the case. And one example that we've been focusing on is actually hemophilia A. So it's a blood clotting disease. And today there is a treatment which basically means that the patients have to inject this missing protein, factor VIII, biweekly into their bodies. You see a little boy who is doing this here. And you can imagine that that is not a very pleasant way to do it. There's a big burden on the patients that have to do this twice or three times a week. And in addition, it is extremely expensive. So luckily, the patients can live quite almost a healthy, normal life span. But of course, the treatment is very expensive. At the moment, it's more than 100,000 euros per year for each patient, which of course is a burden on society because of the costs. And what happens in many of these patients, and again, the movie unfortunately are not playing, but basically what happens is that a piece of DNA on the X chromosome is turned around in these patients. So there's a sequence that is repeated twice, and it happened by chance, basically, that the sequence was turned around, and we've made an enzyme which can basically correct this again. So it recombinase that binds to these sequences and turns this sequence into the right orientation, and thereby is able to correct this genetic defect. We've moved this quite a bit forward. So what I'm showing you here is that we've obtained a sample from a donor, from a hemophilic donor. We made induced pluripotent stem cells from this donor and differentiated these cells into endothelial cells, because these are the cells that actually make the factor VIII. We've then introduced the recombinase into the cells of this donor. And what you can see here in these little bands, the gel here, is basically that only, so you see wild type here, WT is the wild type, these are control cells. These would be cells from you if you're not hemophilic, where you get a band basically when you do a PCR reaction. And only the F8 cells, when you have treated them with a recombinase, you get this band, showing that indeed we can correct this inversion in these cells. And not only that, but we also get expression now again of the factor VIII gene. So we can basically show in vitro now already that we can correct this genetic disease, and that leads to the correction and the production of the missing protein in the cells. Okay, so that presents a very versatile platform that one can use for advanced genetic genome surgery. And I've shown you that you can do deletions and inversions, but also replacements actually are possible. So if you have two recombinase systems, for instance, you can also replace a piece of DNA, which makes this a very versatile platform. But one of the limitations that we are still having is that it's still very demanding. I didn't show you any of the details for time reasons, but actually making a new recombinases by directed evolution takes a lot of hands-on time and also time. So we need many generations of new enzymes to breed them back and to breed them again, to optimize them and so on. So it is a time-consuming process. That's, of course, a disadvantage of this technology, where CRISPR has major advantages, which is much faster and much easier to do. So it's demanding, but we have made already more than 80 different Cre-like evolved recombinases, all targeting different sites. And the power of this directed evolution approach would be shown again in this little movie if it would be running. So we've made 80 different Cre-like recombinases, and what this image basically is showing here is the blue regions are amino acids that never changed. Basically, they were conserved, so we've never seen them changing. But any other sequence that you see here has changed in at least one of the evolutions. And it basically, you find it all over the protein that changes are happening. And you can see some are red. And these are positions where we see that different amino acids they are changed depending on the target site that we're using. So these are hotspots that really seem to be determining the specificity. And what we're doing right now is we're trying to leverage on the information that we're getting. We're sequencing all the libraries that we have for the recombinases that target different sequences. And we are at the moment working on, hold on, trying to go forward. We're trying to use artificial intelligence-based predictions to speed up the generation of new recombinases. So we have all these different libraries of recombinases that target different sequences that we have evolved. We can sequence them. We can sequence the recombinase sequences, and then feed that into an artificial intelligence model, which then is supposed to help us to then basically find, for a new target sequence, already predict the right amino acid sequence for the recombinase. And we've made first steps, and we have first algorithms, and we think they are working. So we are very confident that we will be able to make these new recombinases for new target sites in a much shorter time period to basically move this technology, the platform, into a more versatile one. At the moment, they are still inflexible in that they are very hard to program, but we think that with this artificial intelligence, we will be able to make new enzymes in a much shorter time frame. And we think that these enzymes will then have very high potential for therapeutic genome surgery. With this, I'm at the end of my presentation. I thank you very much for your attention. I have to acknowledge in particular Joachim Hauber from the Heinrich Bette Institute in Hamburg. They are a long-term collaborator on the HIV project, and Maria Teresa Pisabaro from Dresden, with whom we collaborate on trying to understand how these recombinases target their DNA and to rationally engineer them. And I thank you for your attention, and we move on.
Dina?
Hello, and thank you all for your great presentations. I think you presented not so easy to understand topic in a single way, and you presented that in a simple way that people that are even not related to research could understand the benefits, the challenges, and the future of this the modification, genome modifications. Thank you all. We greatly appreciate your contribution. And let's go now to the interactive part of this session. We received some questions from the audience, and let's start with the first one. The first question is to all speakers, if you consider that viruses, bacteriophages are alive. I don't know if John would like to start with this
Oh yeah,
question.
sure. I mean that's a very good question, and I mean we are not going to solve that in the 15 minutes that remain. It's a kind of a philosophical question. I've had lengthy discussions about this topic. Personally, I use the definition that since they have no metabolic capacity, they should not be called alive, since they rely on host organisms to proliferate. They should probably be not called alive. But I also know people that say, well, they are alive, and it's just the virus stage can be considered also or you can compare that to a spore of a bacterium, for instance. So it's a dormant state, and when it enters a cell, it kind of gets active again. So it completely depends where you draw the line, and I think well, it's a very interesting question, but as said, we are not going to solve that today.
Okay, thank you so much, John. I don't know if Mark, Martin, or Frank would also like to add something here, or we can pass on to the second question. Okay, so another question is that gene editing is good for editing, i.e. small changes. And in organisms where it is possible, homologous recombination allows larger DNA sequences to be inserted into the DNA, e.g. a transgene. Do the speakers think these kinds of gene editing tools will one day allow such types of modification? Who would like to answer this question? Martin, please. Thank you.
Perhaps I can take a stab at answering this question. So it is true that sort of insertion of larger or longer sequences into the DNA is something that's a bit of a challenge for CRISPR-Cas systems, CRISPR-Cas-based technologies, but it's not impossible. I think there are many instances where this can be achieved, in either in specific cell types or using kind of specific delivery strategies. What is clear, however, is that the technology it's not a universal solution for all types of modifications yet. But I do think that with with some of the new technologies kind of emerging, or new molecular mechanisms emerging, we will be able to reach a stage where essentially even these kinds of modifications can be done very efficiently and also very very specifically. So I think the signs are there, but the technology is not, let's say, universally applicable in all cell types or all organisms.
Mm-hmm. Thank you. Next question is, how did phages use this system initially? Who would love to reply to this question?
Well, I don't.
Continuation of the previous question.
Yeah, I think it is related to the previous one.
Maybe we can go to the question. When do you think hope each of these technologies will be available for treating people, and how do you tackle the ethical concerns?
Mm-hmm.
Okay, again, I think this is a very this is a great question that I think doesn't have a very simple answer to it. Right now, there are already a number of basically genetic therapies being developed based on CRISPR-Cas. Some of them are already in clinical trials. The aim of most of these therapies is to basically do somatic cell genome editing, so basically target the cells in the body of a patient that basically do not get passed on and will not therefore pass the genetic modification on, because genetic modifications of germline cells are something that is currently not possible, at least legally in many parts of the world, and there's a strong consensus against germline genome modification in humans. But these sort of therapeutic applications are coming up, and we will basically see increasingly clinical trials being conducted, and hopefully in the next few years get to a stage where some of these therapies will be approved for therapeutic use.
Yeah, maybe I can jump in there and extend a little bit. Of course, we see already that actually they are already used as we just heard, and they are in clinical trials, these technologies. I think an important part is, though, that these things need to be discussed, and I think you also mentioned this earlier, that we have to, the ethical aspects, of course, are what many people talk about as well, because it's true, we have basically our own evolution in our hands, and this is, of course, can be used for something very, very helpful, and to avoid suffering, and to cure so far totally incurable diseases. But of course, it also comes with a lot of responsibility, as we've heard before. So we could change other things, right? And I think we need a real discourse in society to basically set the boundaries and say where one can use these technologies for, and where one should be more careful in using them. Germline editing was just mentioned, is of course one. It's not necessary at the moment. We need to find out how safe these tools can be used. We don't want these tools to be moved on to the next generation. This is why germline editing is out of question for almost everyone at the moment. That's, I think, an important aspect of it.
So maybe I could add as well that one framework that might be kind of a good way this can go forward is what happened in the UK with mitochondrial replacement therapy, or what's called 3-parent families. This is where your mitochondria, the energy organelles of your cells, have a small amount of genetic information in them, and sometimes that can become mutated. And the UK Parliament discussed and allowed research to go forward into these therapies, which essentially is a germline therapy, but it's a germline therapy of a very, very small subset of your genes, just the ones in mitochondria. And that process took a long time. It took at least 10 years, I think, of public consultations, expert panels reviewing the data and reviewing how safe the technology was, culminating with a discussion in Parliament, and then legislation that allowed certain centres to carry out those techniques. So I think there are examples of kind of good practice for how this works, but of course all you need is another country to then go ahead and just do it without any legislation. So I think we do need to think about this both locally and worldwide in terms of how we do this.
John, do you want to add something?
Yeah, well, I want to maybe comment on the remark of Frank, where to set the boundaries. That's of course very very important, and we all have to think about that. And I guess many people will have different decisions on where to put that boundary. But I think when it comes to examples where we are repairing genetic diseases, I think there will be very little people that would object to using the technology that at least for certain repair actions is very close by. As Martin already said, I think we should just go ahead with that and do it, or that's at least my opinion. But of course, when you stretch it much further, and then people start talking about design babies. Well, first of all, I think technically that's still very far away for the more complicated features such as talents or whatever. But when it comes to the color of your eyes, I think it's doable. But I think it's not. Well, for myself, that's a step too far to do it like that. And I always say when journalists bring this question up, then I say that if parents find that very important, or one of the parents find that very important, they should have taken that into account when they choose their partners, and not do it using this technology.
Thank you, John. In the meantime, another question arrived. Can these techniques allow to find a cure even for aggressive cancers like brain carcinomas, which are difficult to cure because of brain barriers? We have 4 minutes left.
I think in principle, yes. You know, cancer is a genetic disease, so that means that there are mutations in these cells which basically cause the transformation. So with the tools in hand, one can repair these mutations. I think here, though, the problem is a different one, as in the question already alluded. I think delivery is a very important one, right? So we need to bring, and this is a very active field of research at the moment, we need to be able to bring these molecular scissors into the cells in the body, ideally. And for cancer, it's complicated because basically you have to hit and you have to find every single cancer cell in the body to then deliver the scissors that can then inactivate the cancer mutations that are causing the cells to be transformed. So delivery is a very important research area at the moment. But there are also many advances. We've seen dramatic advances in bringing these molecular scissors into the cells that we want within the body. So I think in the future we will see a lot of progress in this field as well.
But also, I think it's important to mention that these tools will probably and unfortunately not be good enough to solve all problems, right? And for sure, they will help to probably the more simple genetic problems to repair those, and but for some targets and indeed that may also relate, or probably also will relate to delivery issues. It's not going to be the miracle trick that we can now use to cure all disease, I'm afraid. So the expectation should not be too high, right?
Thank you both. Last question arrived. Maybe Mark would like to reply. That phages use CRISPR to bypass the immune system. Is this via adding the methylation, or was this a different part?
It's the bacteria that use the CRISPR to evade the bacteriophage. So maybe there's slight confusion there. But I mean, we're talking, of course, about the bacterial immune systems, not the immune systems of vertebrates. So we use this terminology sometimes very freely in this field, and we talk about bacterial immune systems, that essentially the adaptive systems of CRISPR, which can have a memory of infection, and the restriction modification systems, which are innate because they can only recognize one particular sequence. They don't have any memory of previous infection. So the methylation is simply a way of protecting themselves.
Mm-hmm. Thank you. Maybe Ino had a question. I think this could represent the curiosity of many members of the audience. What is the CRISPR research contribution to COVID vaccine search? If you could, we have 1 minute left. If you could summarize a bit or present your thoughts.
Well, maybe I can say something about that. So in terms of fighting the virus, I don't think that or I have not heard of any idea to use it for that, but for diagnostics, for sure. So the Cas12 that Martin mentioned, but also Cas13, that's another variant, and also the system that appeared on one of Martin's slides. So that's a more complex cascade-like system of type 3. That is all being used now for developing diagnostics to at least detect viral infections, but really to protect people. So to like a vaccine, that's as far as I know, that's not working.
Well, I've seen work based on Cas13 also. So there are Cas versions that actually cleave RNA. There have been, I've seen some work where they actually then try to deliver the Cas13A and to basically cleave the viral the viral, the coronavirus itself. And that's been thought of as a therapy, but I think this is still at a very early stage.
Right.
Thank you all a huge amount for all the work that you put for this session. Thank you to everybody for watching. And the ERC has another 10 sessions, I think. You can watch some other brilliant ERC grantees. So hoping that maybe one day you can do germline modification to produce more scientists like you. Who knows? Anyway, thank you very much, everybody.
Thank you, everybody.
Thank you.
Thank you.
Bye-bye.
Bye.
Bye.